So thank you. I'm glad to see so many people interested in NK cells. And like Katie said, it's a 2-to-1 ratio in the talks here, but she lent me some slides, so I'm going to do my best to even it out. So, and as she mentioned, I did spend 15 years at Minnesota and just moved to Fate, so I guess that's my disclosure for you. And I'd like to just start with a slide to thank all of the patients and their caregivers who have participated in all the clinical trials that we're talking about, because some of this very novel, innovative therapy is a little bit scary. And this is Dan Kaufman, who many of you may know, and Dr. Sandeep Patel with a patient who got the first ever off-the-shelf iPSC-derived cell product. So I'm going to talk you through the history of how we got to this point and then just sort of end up back about the new day of off-the-shelf therapy. So just some more background about NK cells for any of you who do not know. Again, these are CD3 negative, CD56 positive cells, and they have several unique properties. So first of all, they secrete, they're stimulated by and secrete cytokines, interferon, TNF. They can induce natural cytotoxicity, and this is mediated by the net signaling of multiple inhibitory or activating receptors. And these include, for example, KIR, NKG2C and D, natural cytotoxicity receptors, and CD16. NKG2D, for example, recognizes MIC-A, MIC-B, and other stress ligands that viral, radiated or tumor cells may express. And then lastly, importantly, unlike T cells, NK cells do not elicit GVH. So this really also, like NKT, I've just learned, makes them ideal for cell therapy. But lastly, CD16 is a particularly important activating receptor in that it mediates antibody-dependent cellular cytotoxicity, and it is the way that all of these commercially marketed monoclonal antibodies work, by pulling in NK cells as effectors to the tumor. So many groups have taken advantage of NK cells for cancer therapy, and as I mentioned, monoclonal antibodies use them as their effectors. And in the human population, there are two variants or alleles of the CD16 or the Fc gamma receptor, and there's a high affinity and a low affinity allele. And only 15% of people are homozygous for the high affinity allele. And what that translates into clinically is shown in this example with the use of Herceptin for breast cancer, that if you're one of these patients that's homozygous for the high affinity allele, you have a much better progression-free survival as compared to those who don't carry it, 2 copies. And there's similar observations that have been made with Erbitux, for example. So one company, Macrogenics, is exploiting this by developing antibodies that are optimized to be used with the 85% of people who are not homozygous for this allele, and this is just an example of a trial where they compared trastuzumab to their optimized agent and showed improved survival. That said, all of these methods take advantage of the patient's endogenous NK cells, but we know that cancer patients have dysfunctional NK cells due to all the tumor suppressive mechanisms that we all know and love, MDSCs, regulatory T cells, et cetera. So what we really need is approaches to restore or replace dysfunctional T cells or NK cells in cancer patients. So what I have is a table that I'm going to go through, which will talk you through the history of NK cell-based therapy over time. And it all started in 1985 at the NCI with Steve Rosenberg with the lymphokine-activated killer cells, or IL-2 activated autologous patient NK cells. And a clinical trial was performed, but it didn't have great efficacy. And part of the reason is the concept of missing self. So NK cells have, as I mentioned, inhibitory and activating receptors, and specifically KIR, or killer immunoglobulin-like inhibitory receptors, recognize inhibitory ligands on normal cells, which happen to be self class 1 MHC. So we tell these cells, don't kill us. So in an autologous NK cell, if you have inhibitory signals and not enough activating signals, you won't get killing. However, if you have a loss of the self-inhibitory ligand or downregulation of MHC, then you will have a net balance that tells the cell to be killed. So loss of heterozygosity or B2M mutations and downregulation of class 1 can help for T cell escape, but then it renders you susceptible to NK cell killing. But in the real life, this is downregulation is variable and incomplete, and that's why allogeneic NK cells are more promising to have a higher frequency of alloreactive cells that are not going to be inhibited by the patient's tumor. So moving on, many groups, including Minnesota, became very interested in using allogeneic peripheral blood NK cells. So at Minnesota, we did lots of trials using IL-2 or IL-15 activated cells. So shown here is a schema starting in 2000 where patients with relapsed refractory AML had a related haplodonor from whom a peripheral blood apheresis was collected. It was CD3 and CD19 depleted, overnight incubated in either IL-2 or IL-15, and then infused into the patients after the same lymphodepleting preparative regimen pioneered by Steve Rosenberg. And then after the cells were infused, doses of interleukin-2 every other day for 6 doses were given. And then we followed for outcomes. And what we found was about 30% of very refractory AML patients could achieve a complete remission as a bridge to transplant. And in about 10% of patients, we could detect a lot of peripheral blood donor-derived NK cells. So we did a lot of trials in leukemia, lymphoma, ovarian cancer, and what we found was that the IL-2 was driving regulatory T cells, and that was sort of counterproductive. So we came across the strategy of trying to use ONTAC, or an IL-2 diphtheria toxin fusion protein, to pre-deplete the regulatory T cells and then to infuse the donor NK cells. And when we did that, compared to IL-2 alone, the ONTAC-treated patients had much better peripheral blood expansion of the NK cells, and that seemed to correlate with better overall remission rates. But unfortunately, that went off the market. So we had to have another hypothesis. So we thought, well, maybe using IL-15 instead of IL-2, ONTAC or IL-2 would be a good strategy. And so again, they both activate NK cells and signal through the common gamma chain, but unlike IL-2, IL-15 does not drive regulatory T cells. So we did 2 trials using the NCI's monomeric recombinant human IL-15. There are other IL-15 agents on the market. Altor has, or now Nant, has a IL-15 agent as well. But I'm just going to summarize our data here. So we first did a trial using the same haplodonors. Instead of overnight incubating the product in IL-2, we incubated it in IL-15. They had the same lymphodepleting preparative regimen, and there was an IV dose escalation dose with the cohort shown here. This was followed by another trial using subcutaneous dosing, and this dose was chosen based on a solid tumor monotherapy trial at the NCI where this was the safe dose. And then we assessed for clinical response. And this is about to come out in publication, but the data are important for a reason that you'll see in a minute, so I wanted to present it here. So what we found was that with the IV dosing, as with every other trial of NK cell adoptive transfer that I have ever seen, there was no CRS and no neurotoxicity. However, when we used subcutaneous dosing, we had quite significant CRS and neurotoxicity. And so we wanted to explain this, and I do not believe it was the NK cells doing it. I believe, as I'll show you in a minute, that we were overdosing them with IL-15. So what happened, shown here is the IV dosing in blue and the sub-Q dosing in red. And you can see that the IV had, a high, fast peak, and then it went down quickly, whereas in red with the subcutaneous dosing, this is right before the second dose. It hadn't cleared yet, and it was accumulating and accumulating because we had failed to consider the role of lymphodepletion as opposed to the solid tumor trial, and there wasn't a sync, and we, I think, just way overdosed the patients with IL-15. That said, the response rates in the IV and sub-Q dosing arms were pretty similar to what we had seen with IL-2 regimens, and it wasn't a dramatic improvement, so a little bit disappointing. So that summarizes the Minnesota experience, and then lots of groups started looking at his umbilical cord blood as a source, because getting a donor in, and it's just very complicated. And so glycostem and then Celgene, now Cellularity, have had trials of these approaches, and one of them, these were presented at AACR most recently. I led the trial for AML in Minnesota. And I think the key point is that there's still a lot of donor heterogeneity. There's still a lot of manufacturing challenges. It's expensive, and the clinical benefit that's worth exploring, but it wasn't a home run. So moving on, the next approach is to try to ex vivo expand a product using peripheral blood NK cells. So this has been done by Todd Feniger at WashU with the cytokine-induced memory-like cells. Dean Lees from MD Anderson now, Ohio's program. Green Cross had some allo NK cells. Gameta has a NAM-activated product, and then Fate Therapeutics made an adaptive NK cell product. So I'm going to tell you about that. So adaptive NK cells are those that you find in the peripheral blood of individuals who are CMV seropositive. And this is shown on this graphical abstract that when you have CMV exposure, you downregulate a lot of these adapter proteins and have epigenetic reprogramming of NK cells. And this subset are very resistant to Treg, to MDSCs. They have memory-like properties, and they have very potent ADCC. So we thought this would be a good effector population to use in a clinical trial. So working with Fate Therapeutics, we, from Minnesota side, developed a strategy to take peripheral blood mononuclear cells from seropositive donors, take out the T and B cells, incubate them for 7 days in a special culture condition, and then have a cell product for testing. And so that was done at Minnesota, same Cy/Flu prep, cells are infused, interleukin-2 every other day for 6 days, and then we looked at outcomes in a dose escalation study. And actually, this is one of 3 trials using this product, one in AML. There's another trial in ovarian cancer, where interestingly the cells are delivered intraperitoneally to try to get to the homing issue in ovarian cancer. And then a Fate-sponsored study in solid tumor with several arms, some of which include targeting with trastuzumab and cetuximab. And there's one data slide I want to show you from this whole experience because I think it's important. So this is showing cells collected from intraperitoneal fluid from a patient who was treated twice. So in the ovarian cancer and the solid tumor trials, we allowed repeat dosing if there was clinical response. And the first dose, what you can see if you gate on NK cells, based on differences in the donor and the patient's HLA, you can differentiate the product or donor HLA from the patient's NK cells. And this is 12 days after infusion, and you can see they're still there. Because she had clinical benefit and was redosed, she got another infusion from the same donor, and now here at 12 days you can see again a significant, in fact better proliferation or persistence of those cells, showing that repeated exposure at least twice didn't lead to alloimmunization. So those trials are ongoing, but again what we've learned is that you have to probably give them with cytokines. Persistence is a problem. Donor availability is a problem. Donor product heterogeneity is a problem. It's very expensive. It's very cumbersome. So the world turned to cell lines as a potential solution. So NK92, NantQuest, and then a lot of work in China has been done trying to optimize these cells for therapeutic purposes. In fact, now we're for the first time looking into genetic engineering and modification of these cells. And I won't show you a lot of data, just to point out one real limitation is the cells have to be irradiated. So they go in sort of actively on their way out of function. So they have a very short functional window. And they can be given in repeated dosing, but it's still a limitation. So here's some references. So the next strategy my wise colleague over here came up with was to try to introduce a CAR into NK cells. So at MD Anderson they did a clinical trial, and Katie lent me her slides, thank you very much, shown here where they introduced the CD19 CAR, and they also engineered in an IL-15. into a cord blood-derived product. So then they were transduced, expanded, and multiple dose levels were tested in a clinical trial, again, after some lymphodepleting therapy. And here really a picture is worth 1,000 words. Of the 9 patients treated on this phase 1 study with very heavily pretreated, relapsed refractory, very bad lymphoid malignancies, you can see dramatic responses. They had 6 CRs, 1 PR, and importantly, no CRS, no neurotoxicity, and no graft versus host disease. So this is, Katie will be talking more on Thursday. Please go if you want to hear more. But this is really exciting and sort of proof of principle that NK cells and NK CAR can have clinical benefit. So that said, I'll speak for Katie saying perhaps it's challenging to engineer these products, it's expensive, et cetera. And so Fate Therapeutics has been developing an iPSC platform for cell therapy. So that is shown here, where they start with induced pluripotent stem cell population, and they're able to do gene engineering on that population to introduce or knock out the genes or CARs or targets of interest. And you end up with multiple clones that can be plated at a single cell level, each of which has been engineered once. And you can grow up each of these clones and then do selection to look at chromosome stability, the efficiency, if you knocked in the right number of copies, if you knocked out what you wanted to knock out, for characteristics such as the potential to grow in culture. And then you can make a master cell bank from which multiple manufacture campaigns can be run. And so what's shown here is you can get to a CD34 stage, which can be frozen down, and then you can, under different culture conditions, grow up either NK cells or T cells for further off-the-shelf cell therapy. So this is really a big change in our ability to make large numbers of cells for true off-the-shelf therapy. And so what's shown here in a little more detail is just the manufacturing process to make NK cells, again, starting from a very, very, very pure, 99.9% pure population of engineered iPSC cells, which you differentiate into a CD34 cell, and then under different conditions can grow up a very large expansion of the NK cells, ending with a product where if you started with 1 e to the 6th iPSCs, you can end up with at least 1 e to the 12th of cells for the clinical trial. So these cells function, which is very important. So shown here are some data demonstrating that compared to target cells only, peripheral blood NK cells in blue, cord blood NK cells in purple, the iPSC-derived NK cells in red have the best killing over time of multiple different tumor targets. And importantly, when these cells are frozen down, the cryopreservation media is actually infusion media, so you can just give them directly off the shelf. So what's shown here is the ability of the cells to eliminate the targets directly thawed. So you take them, you thaw them, you infuse them, versus needing to resuscitate them and put them in cytokines over time, compared to immediately defrosted, for example, peripheral blood NK cells, where they don't function very well without a rescue. So the first iPSC NK cell product is called FT500. It has no engineering as per the FDA's request to check the safety of this without any manipulation. And so the product was manufactured at the University of Minnesota in their GMP facility. And they were able, after this 44-day process that I showed you, in 1 day to produce hundreds of cryopreserved bags at the concentration needed for the clinical trial, which is clearly much less expensive than doing one product for each individual patient. And shown here are the release specifications, and this product has high levels of NKG2D, NKP30, the cytolytic proteins, granzyme and perforin are at very high levels, and low levels of checkpoint expression. So this is what the product looks like, and it's just ready to go. You ship it to the site, they thaw it and infuse it within 15 minutes. So that was very exciting, and the trial to study this is a 2-arm study testing it as monotherapy for a safety evaluation, and then in combination with checkpoint blockade for patients who have advanced solid tumors with an intent to enrich for checkpoint-resistant patients. Why is this rational? It's because one of the significant mechanisms of resistance to checkpoint is this loss of class 1 expression or B2M knockout. So as you lose your class 1 and escape T cell recognition, that, remember, renders you susceptible to NK cell-mediated killing. And this mechanism appears to be important in about 30% of checkpoint failures. So that's a really good opportunity for an NK cell to come in and try to rescue those. So this is the clinical trial schema. You can see in cycle 1, they get a lymphomodulating preparative regimen as an outpatient. They get 3 weekly doses in a dose escalation manner of the product. If they're on checkpoint, they continue it. If they failed it, they would continue the one they failed on. And then at the end of 28 days, if there's been no DLT, they can go on and get a second cycle of 3 additional doses, after which time we evaluate for response. And so the first cohort of monotherapy has been completed. So the first 3 patients were treated. You can see here their diseases. And patient number 1 actually got all 6 doses and is waiting for his disease rescanning. And the other 2 patients got 3 doses with no DLTs, no SAEs, and they're continuing. So that means we're open to dose cohort 2 for monotherapy and to dose cohort 1 with the checkpoint arm. And then the other trial I want to tell you about is the first engineered product. So I mentioned that CD16 is a very, very potent activating receptor mediating ADCC. And in real life, CD16, after it mediates ADCC, is cleaved by ADAM17, perhaps as a regulatory mechanism to prevent overactivation. So what has been engineered into what was the FT500 cell is now a non-cleavable or cleavage-resistant high-affinity CD16. So this is that high-affinity variant that only 15% of individuals are homozygous. So this will be a much more potent activating receptor. And so this is the product profile, again, just showing the purity that you can get of the product when you do the engineering at the clonal level and then grow up all the cells from there. So this FDA-approved trial has 3 arms. There's a monotherapy regimen for AML, and then there's combination with Rituxan for lymphoma and with ELO for multiple myeloma to see if we can exploit this super CD16 receptor. And so the schemas are shown here. Again, there's a outpatient lymphomodulating Cy/Flu regimen followed by 3 weekly doses of the cells. In this trial, we are giving it with IL-2, and then depending on which disease, here's the other therapy that goes with it. So it's a similar schema to the solid tumor trial. So to summarize, I think that NK cells are a very attractive effector cell for cancer therapy because they do have some unique properties for unique settings that'll be important. So how can we address the remaining challenges and realize the potential of this therapy? So certainly the cells have to function. So we want to try to enrich either through culture, methods in some trials or by genetically engineering to try to get high expression of activating ligands, metabolic profiles, etc. And we can now induce specificity and targeting by putting in CARs or by targeting with bikes or trikes or monoclonal antibodies exploiting CD16. Or you can engineer in IL-15 as Katie has done with the NK CAR. Now we have a way to address antigen escape or loss of MHC by putting in dual CARs or by remembering that NK cells have these native activating receptors that recognize sick tumor stress ligands that don't require such specificity. Homing, I think, for hematologic malignancies is less of an issue. That's where NK cells are in their sweet spot. But over time, understanding how to guide them to solid tumor beds and what chemokine receptors, et cetera, are going to be ideal, I think will be important. And then we want to look at dose optimization and persistence, and how do you get sort of an area under the curve exposure of the tumor to these effector cells. And that can be done with repeat dosing or by using engineering techniques to engineer stealth, perhaps taking out HLA, MHC, et cetera. And then lastly, we are starting to address the scalability and the cost and the heterogeneity of all these processes by having engineered clones, and for the first time really a true off-the-shelf therapy that you thaw and give in an outpatient setting. So with that, I would like to just acknowledge all my colleagues at University of Minnesota, Dan Kaufman and Dr. Patel at UCSD, and then Fate Therapeutics. Thank you.
Well, thank you, Sarah, for a really outstanding overview of the field and exciting new data. We have time for a couple of questions. Yes.
I was wondering if you could speak to the phenotype of the differentiated NK cells, and in particular their maturation state. Have high CD56 expression. They look like mature, not immature bright cells, but they look like mature, fully differentiated cells, if that's what you're asking.
Yes?
Just to complete your table, what do you think are the limitations for the iPSC?
Yeah, that was sort of my last slide. I mean, I think it's just about making sure that they can persist, thinking about mechanisms of host rejection, and can you overcome them with repeated dosing or engineering and stealth, homing to their tumor beds as we're looking into solid tumors. And I think those are the things that I worry the most about.
Yes.
Two quick questions. Giving IL-2, does that introduce liability in your beta program? And then secondly, the antibodies, Rituxan and the other one, what kind of delta are you looking for?
So IL-2, many NK cell trials have used IL-2. And in the FT500 trial, there will be no IL-2, so there's no cytokine support, as opposed to the hematologic malignancy trial where we will use IL-2. So I think comparing across those two trials, we'll learn if it's important. It's pretty safe in all of the experience to date of using peripheral blood or other NK cell adoptive transfer in heme malignancies with IL-2 has been safe. So we'll just have to see. And the other question was about Rituxan. I think at this point it's a phase 1 safety trial. We're just looking for responses. The patients will have had to be refractory to be eligible, so just any sort of clinical response in a highly refractory patient would be encouraging. The trial's not really designed to look for a delta yet.
So Sarah, last question. You mentioned with the iPSC-derived NK cells, you've got the advantage of being able to pick the clones. What do you mean by that, and how do you pick these clones?
So you do your engineering of interest on a batch of the cells, and then in a single cell manner, you put one cell of each of the clones, and then you grow up a population. And that population then can be studied for the properties of interest, so ability to grow well in culture. Did the gene editing happen into the right place? Do you have the right number of copies? And then once you've picked the optimal clone based on, phenotype, function, growth, then that's what you make your master cell bank out of, and you can freeze that down and then do multiple manufacturing campaigns from that.
And with your 44-day, you said it takes 44 days to grow. These cells at the end, they don't have any properties of exhaustion, or when you transfer them into animals, they don't become exhausted?
At this point, no. And so I didn't show it, but when you put them into an NSG mouse, they will persist. They persist longer if there's some cytokine support. But then in the patients, there will be the lymphomodulating regimen that does induce IL-15, et cetera. Or you just engineer in IL-15 down the road would be the strategy.
Okay, great. All right, so if no other questions, thank you everyone for a fantastic session. Appreciate you all coming.